In this final video regarding the metabolism of glucose, we have glycogen metabolism. So the building of glycogen and the breaking down of glycogen. And so by the end of this, I want you to be able to explain the difference between how amylase and phosphorylase function and its significance with regards to glycogen metabolism, to describe the function of phosphoglucomutase, and describe the process by which glucose molecules are activated for glycogen production. Now, dietary starch, starch that you get from your diet, can be broken down in one of two ways. And the first is through something called amylase. Now, amylase is an enzyme that will use water to break down our starch or glycogen. And in so doing, the direct byproduct or the direct product of this reaction is simply glucose. Now, phosphorylase, however, so here is our phosphorylase, again, taking endogenous glycogen. So this is glycogen not from our diet, but glycogen from our cells. And phosphorylase uses something called phosphorolysis. A phosphate group is used instead of water to help break off the glucose molecules from the glycogen. And the direct product of this is a phosphorylated glucose molecule, glucose 1-phosphate. And so if we look at that reaction a little bit more closely, what we see here is that our glycogen or starch phosphorylase uses this inorganic phosphate that's floating around. Notice it's not coming from an ATP molecule, and it attacks the bond between the glycosidic bond between the last glucose of that non-reducing end of glycogen, and then therefore the product being glucose 1-phosphate. Now, the problem is glucose 1-phosphate is not an intermediate of glycolysis or an intermediate of glycogen synthesis if we were going to use it to make glycogen. So the question is, how do we convert glucose 1-phosphate into an intermediate of glycolysis? We use an enzyme called phosphoglucomutase. And this enzyme, and it's all in blue, is our enzyme itself. So this serine is part of the enzyme, and it starts phosphorylated. So our enzyme starts phosphorylated, and what it does is when glucose 1-phosphate enters its active site, the enzyme phosphorylates glucose carbon number 6. So now there's a very transient intermediate of glucose 1,6-bisphosphate. But remember, an enzyme always has to end the way it started, and so now what happens is the enzyme grabs the phosphate group from the 1 carbon, leaving us with our final product, glucose 6-phosphate, and we end the enzyme the way it started with a phosphorylated serine here. Okay, so glycogen biosynthesis requires something called a sugar nucleotide. And a sugar nucleotide is our glucose that has been activated by a NDP, a nucleotide diphosphate, in this case a uridine diphosphate. So if we are going to build glycogen out of glucose molecules, we need to start with glucose 1-phosphate. So how do we get to this? Well, we take glucose 6-phosphate out of glycolysis and we use that mutase to turn it into glucose 1-phosphate. At this point, we use this enzyme called NDP, sugar pyrophosphorylase, and what this does is it takes here's either our UTP or a GTP, right? Some sort of nucleotide triphosphate. Our sugar then, that our phosphorylated glucose, attacks the first phosphate group of the NTP, and that leaves then pyrophosphate as our byproduct, and we get our sugar NDP sugar molecule, our activated glucose. Now, pyrophosphate is then broken down again into two phosphate groups. That's a very exergonic reaction. And so what we have is a sort of a coupled reaction here. We have the activating of the sugar as one reaction, and then the breakdown of pyrophosphate as the second reaction, and the two then are coupled. Now, this NDP sugar is important because this giant NDP part of the NDP sugar is a great leaving group. It very much increases the reactivity of this sugar molecule. It also serves as a tag for the cell to determine, okay, great, this sugar is bound to be added to a glycogen molecule. Now, the way that happens is, here we have our growing glycogen molecule. This is the non-reducing end, so that's our carbon 4. The hydroxyl group of carbon 4 is what's going to then attack the anomeric carbon of the activated sugar. And this whole UDP is going to be our leaving group and our byproduct. And then we have our, using the enzyme glycogen synthase, we now have an added molecule of glucose to the glycogen. Now, make sure you watch this video again. Make sure you can explain the difference between how amylase and phosphorylase function and its significance. Describe the function of phosphoglucomutase, and describe the process by which glucose molecules are activated for glycogen production.